This week, we are learning about lake ecosystems. Limnology is the study of freshwater biological communities and their interaction with the surrounding chemical and physical environments. Lentic ecology specifically refers to deep flow moving waters like ponds and lakes. We're going to be studying Peter Lake, which is located in the Northern Highlands Lake District in Michigan. It is important to study lake ecosystems not only because of the aquatic life they support, but also because of the link they provide between terrestrial and marine ecosystems. Carbon inputs from land, such as leaves or decaying leaf matter, are important for primary production and microbial activity in aquatic food webs. Carbon can affect light attenuation, which has implications for primary producers, and can also contribute to acidification of poorly buffered freshwater systems. Buffering capacity of a system simply refers to its ability to resist changes in pH in response to the addition of acids and bases. And this is related to the salts and minerals present due to local geology. As you can see from this diagram, a lot of carbon processing and storage occurs in lakes and rivers. There are a number of different methods for monitoring changes in aquatic systems, but they are usually expensive in terms of time and money. Sondes, on the other hand, pictured in the upper right corner, these are relatively inexpensive and they require little care and generate heaps of data. Sondes are devices that are deployed and passively take measurements on physical and chemical parameters such as temperature or light levels at a given time interval, so every second or hourly or even daily. In this lab, we will be examining hourly measurements of PAR, or photosynthetically active radiation, at the surface, and temperature, dissolved oxygen, and pH of the water column from a sonde that is deployed in Peter Lake. In particular, we're focusing on diurnal patterns, meaning patterns that occur throughout a 24-hour period. The graph pictured here is based on temperature data from the Peter Lake dataset you will be working with. A chemical parameter we will be considering is dissolved oxygen. Dissolved oxygen simply means the amount of molecular oxygen in water, which can enter by diffusion from the air, which is increased by turbulence such as wave action, or as a byproduct of photosynthesis by submerged aquatic vegetation or phytoplankton. Note that dissolved oxygen is consumed by organisms during respiration. Because the solubility of oxygen decreases with increasing temperatures, when radiation from the sun heats the water, rising temperatures throughout the day will affect dissolved oxygen levels by changing the direction of this diffusion that occurs at the air-water interface. For your first assessment, which you need to bring to class, draw a figure of what you think dissolved oxygen dynamics would look like over a 24-hour period if, in scenario A, only temperature was controlling dissolved oxygen, and in scenario B, metabolism is the main control on dissolved oxygen. And metabolism is just referring to photosynthesis and respiration. Another chemical parameter we're interested in is pH because of its relationship to photosynthesis and respiration. Because Peter Lake has a low buffering capacity, the system is sensitive to changes in pH due to the metabolic functions of organisms in the lake. So when carbon dioxide is consumed during photosynthesis, the system becomes less acidic. When carbon dioxide is produced from respiration, the system becomes more acidic. Thermal stratification is a characteristic of some lakes that contributes to differences in dissolved oxygen and pH at different depths. Thermal stratification occurs when energy from the sun heats the surface of the water, creating a warmer, less dense layer called the epilimnion. Cooler, denser water sinks, creating the hypolimnion. The middle layer, the thermocline, is a layer in which temperature decreases rapidly with depth. Some lakes may remain stratified year-round, while some experience turnover due to changing temperatures. For example, Peter Lake likely experiences summer thermal stratification, a fall turnover, reverse stratification in the winter, in which a layer of ice floats on the surface and warmer water sinks, and spring turnover. Turnover events are important because mixing occurs between the layers, allowing for dissolved oxygen and nutrients from the bottom to reach the surface where most photosynthetic activity occurs. So, during a summer day at Peter Lake in the epilimnion, you might expect to find higher levels of the oxygen and lower nutrient levels as they are quickly assimilated by producers. And remember, in this layer, during the day, production is greater than respiration. In the hypolimnion, you might expect to find low dissolved oxygen because it's consumed by microbes involved in decomposition and thus higher nutrient levels and a low pH. In this layer, respiration exceeds production during the day. Remember that the bottom waters are more acidic because respiration exceeds photosynthesis and there is a greater amount of carbon dioxide. Consider how these patterns might change at night when there is no sunlight to drive photosynthesis and temperatures drop. The goal of this lab is to understand how we can interpret patterns that we observe from the chemical and physical data and then understand changes to the system over time.